We started, yay! Right, a minute on time. So let me review, as is my custom. Ah, so much better. This is, we looked at that animation, and really, it's the whole idea is this summarizes all of these events at the neuromuscular junction. They have this done a couple of different ways, just to go over it without doing the animation again. Effectively, the impulse you create, you decide to move something by way of the voltage. Basically, it gets through the nerve, and when it gets toward the motor end plate at that synapse, at the neuromuscular junction, before anything can happen, it triggers voltage gated calcium channels in the distal portion of the nerve to release calcium. Same thing when nerves converse with one another. The calcium will enable or facilitate the exocytosis of the synaptic vesicles, which contain the neurotransmitter. In this case, the neurotransmitter is acetylcholine. That's significant. It rapidly diffuses in such an infinitely small space, even though it's a space. It will trigger, they call them sodium chemical gates, but really these are channels that allow sodium influx and potassium efflux. So, I mean, if you want to, I would consider them to be sodium potassium channels. That's how I would describe them. But really, what we must understand is they're ligand or chemically gated channels. It's just the presence of the chemical, and as long as it is there in the synapse, it remains open. Sodium floods in, potassium floods out, more sodium, 3-2 relationship. So it's always going to be excitatory. It's going to take that resting membrane potential, bring it closer to threshold. We'll get into that a little bit as soon as I finish this review. And then what happens after that, whether or not it gets to an action potential, we'll consider that next. But for the time being, the acetylcholinesterase, which is very, very rapid, things that occur rapidly go away rapidly, consistently within the body. That's part of physiology, almost always the case. These are rapid channels because they flow ions in and out. So they're almost an electrical type channel, if you will. So the acetylcholinesterase is always there, degrades it, and interestingly, it does all three ways. The acetylcholinesterase does the breakdown or metabolism. The acetic acid is used elsewhere and diffuses out. The choline, they talk about reuptake. That's the choline is brought back into the nerve to be resynthesized into more acetylcholine, and whatever is not done that way to remove it diffuses out. So all three methods of removal of neurotransmitters, generically speaking, occur there. Why it's a good model. They go over all this for you to look at. I'm not going to review it again. You don't need to. The key is here, leading to what today's topic is. The sodium ions enters, then potassium exits. The membrane, it's called an end plate potential. It must reach threshold. Threshold is this change of sufficient amount of voltage to reach some arbitrary number, roughly from minus 70 to minus 55 average. And at that point, effectively, it's really what you will see shortly is how far that change travels, because it degrades rapidly, as you've just seen. And so, if it is not sufficient to reach a significant series of landmarks, you will come to see those landmarks are sodium, potassium, voltage gated channels in sequence. And once you hit them, then that impulse, the rapidity of triggering and re-triggering channels in all directions, it isn't unidirectional as you see with nerves. It goes wherever if it's stuck here in the middle of my arm, that impulse goes this way, that way, forward, backward, side to side, wherever. So it's all over from and effectively that whole segment of that muscle, that whole fiber and the related structures will fire. That's the beauty of that system. So if it doesn't reach what's called an action potential, and it's just trending towards it, we call it an end plate potential. That's an important distinction why I'm making it, because when we get to nerves, we're going to look at a different type of nomenclature because sometimes they're excitatory and sometimes they're inhibitory. So terminated by acetylcholinesterase, diffusion, et cetera, and we did the animation. So a lot of this, there are problems. So this is a common area. I mean, for instance, this is something that I mentioned before to you. Botulism toxin certainly impedes this area. Tetanus toxin does not. That's where the spinal or cranial nerve meets up with the motor nerve going to the muscle. Okay? So Botox impedes the release of, by way of the synaptic vesicles. It doesn't allow that fusion of the synaptic vesicle and exocytosis. It precludes it. The classic example would be myasthenia gravis. Okay, characterized by, you see, as we get older, it begins to manifest itself. It becomes typically the reason why we see it swallowing, talking, and particularly drooping eyelids before generalized muscle weakness. It always affects the muscles you use the most, and your eyes are moving all the time. Your eyelids are constantly moving. So they're the ones that are fatigued most. I've had a couple of patients with this. Thankfully, there are kinds of treatment that are available. So this is a shortage of acetylcholine receptors because they are being attacked. It strongly suggests it's an autoimmune disease. It's there. There is treatment to try to prolong the amount of acetylcholine in the area. That's another issue in and of itself. So, in general, resting sarcolemma is polarized. We know what that is. Distinction across the membrane, not in the interior or exterior, but directly across the membrane, more negative on the inside, positive on the outside. We did this endlessly. So the action potential starts with an end plate potential. If it reaches threshold, then you will see the events after that are very rapid because these voltage-gated sodium and then potassium channels very close to each other, abundance of them, and very, very rapid transition. So you will rapidly see depolarization. You'll see it graphically in a second, and repolarization. Depolarization, rapidly shifting it from positive on the outside, negative on the inside, to the opposite. Negative on the inside, negative on the outside, positive on the inside. And then repolarization means to reverse it. And so what happens, what depolarizes, because there's a rapid influx of sodium, and as soon as that happens, then there's a rapid efflux of potassium by the potassium voltage-gated channel, so it's, and that's that waveform. And in the meantime, back at the ranch, right away, those sodium and potassium pumps are going crazy. So using your muscles, as we all know, or your nervous system expends a tremendous amount of energy. That's why I guess I always see students in my lectures falling asleep, because they're thinking so hard about what I'm saying. It can't be because I'm bored. And I try to inject all the jokes, funny humor, clever pictures of my attractive grandchildren and children. Yet, well, you're awful. How's Sadie doing? Abby, how's Sadie doing? You hear from her?
Yeah, she's doing well.
Good. She's in Columbus?
Yeah.
Cool. She would have laughed. That's why I asked. At least I had someone who would respond. The rest of you, don't take it the wrong way. You're coughing but smiling at the same time. Don't like anybody. End plate potential. Acetylcholine released, binds, chemically gated ion channels, sodium diffuses in, potassium diffuses out, but not much. Okay, more positive, less negative. End plate potential. So it has to be an end plate potential to start. Okay, is generated, and with the point they're trying to get apart graphically, the change is localized, and because the acetylcholine is removed so rapidly, if it cannot extend far enough to threshold, and threshold will take it to the next area adjacent to, but not as close as you might think, to the end plate itself, because that's those sodium and then potassium voltage gated channels. So depolarization, it's generating it and propagating it, continuing on in all directions. If it causes enough change at the end plate to reach that threshold, then it has traveled far enough to the voltage gated sodium and then potassium channels. At that point, we call it self-amplifying. It just continues. At that point, those generate a very large influx of sodium, triggering the action potential that is, dare we say it, unstoppable, and will lead to muscle fiber contraction. The action potential spreads across the entirety of the sarcolemma, from one channel to the next in adjacent areas, causing that. So just the very fact that it's changing from positive on the outside and negative on the inside and flipping it induces the next area and the next area. So one change induces the next change and the next change until it reaches its termination. So voila, that's what you're trying to show you here. Again, that what we call the inactivation gate is not really relevant in this discussion. So again, sodium enters following its electrochemical gradient. At a certain voltage, an action potential is generated. Action potential spreads adjacently. Sodium channels there, propagating, etc., like ripples. Repolarization. Understand what we're doing here. This is your electrical events. Sodium in is changing from positive outside. Now it's negative on the outside, relatively speaking, positive on the inside. That depolarizes. And so these are electrical events mediated by the rapid transiting of these charged particles, ionic particles. Again, we have to reverse that. So we're going to reverse the electrical event. That's repolarization. Then we have to bring it back chemically, and that's what the sodium-potassium pumps do. They restore the original ionic concentration, pumping back in the potassium and pushing back out the sodium. In the same relationship, 3 to 2. And they'll show you that. So you can see the rapidity of it. So here, roughly, if this is threshold, whatever, arbitrarily, wherever, they don't even necessarily have to have the number. You can see this spike is well over a millivolt. You would do well to remember that when we start to look at the heart. We see this very distinctive spike that's there. Okay, and you can see the time elapsed really from when it fires into when we're talking about, less than 10 milliseconds. So you could, 100 of these a second could easily occur. Not without a price to pay over time. And you should keep that number in mind when we start talking about basically different patterns with regard to the muscle itself. What this is all about is this EC, or excitation contraction coupling. This is the excitation. We have to somehow couple this to the activity of causing the filaments to slide over one another. So we excite them, and then we have to develop the contraction of sliding. By propagating that action potential, you're going to see graphically how the T, or transverse tubules, these interesting invaginations that enter deeply into the muscle fiber and surround those fibrils, are going to trigger voltage sensitive, here we go again, calcium channels in the sarcoplasmic reticulum at those aforementioned triads we talked about, where the terminal cisternae on either side meet the T, or transverse tubule, and will generate this action potential that's brief. And you can see electrical events consistently precede mechanical events. The action potential, brief and ends before contraction. That's a really pivotal point, and you need to recall it because it really becomes significant when we start looking at cardiac physiology. It's not quite a skeletal muscle, but the concept is definitely there. So this is what it looks like, the sequence of events along the sarcolemma, and it's better to see it graphically than with these illustrations like this, and like this, and like this. So I'll defer to the animation as we speak. I will likely have to adjust the volume. Bear with me and listen.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma.
You see, this gives you a pretty good view here of how they surround the individual fibrils. These are all the T tubules right along that A and I band area. So, I mean, it really gives you a good look at what's going on, I think, from this animation.
And is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
To me, that it really shows. It shows the extensive nature of the transverse tubules, which again are part of the sarcolemma, the plasma membrane. The extensive network of endoplasmic reticulum, the sarcoplasmic reticulum, all smooth and a ton of calcium storage, and there's a bunch that's in there. And because I mean, it takes a while to get it all out. That's why you can sustain muscle contractures and increase them dramatically with the amount of calcium that's stored in there inherently and available, let's say. And then you can see again, it started with the nerve to get that nerve to fire, a voltage-gated calcium channel, and it's going to end this part by triggering a voltage-gated calcium channel at the juncture point in those triads to release the intracellular calcium, which is stored in the terminal cisternae. It's really remarkable when we see this. So I mean, I really like those, and I mean, I go through these rapidly because I can't read them, and I figure that the writing is so hard, but it's basically telling you the same thing. The start of those channels, once it hits, you're starting right here where threshold is generated, just looking linearly here or here, but in all directions, as I said. And then you can look at the idea. So excitation, this is basically the coupling of this wave with the action potential to the actual events. And it's that calcium flooding inside the cell, the intracellular calcium being released from storage and now flooding the interior cytoplasm, or in this, the muscle cytoplasm, that we call sarcoplasm, that will create this interesting chain of events that we're going to look at next that are there. And that's the chain of events is going to be this part. We're going to look at troponin and tropomyosin, the calcium attaching to the troponin plugs. They're changing shape, molecularly speaking. That change of shape facilitates the movement of the tropomyosin threads, exposing those binding sites on the actin, the myosin binding sites on the actin, to actin binding sites on the myosin. The only reason I can do this so easily is I've been doing it this way for the last 25 years. So I'm not expecting you to know it overnight so glibly. Is that a word? Yeah, with a glib. and voila. And then we're going to see that pivoting agent with as they attach to the myosin head, it pivots, ratchets, whatever word you want to say. Fires is a good word for me. And then pulling it toward the midline, causing the sarcomere to effectively shorten, even though the structures within are not shortening, they're sliding over one another. Hence the name sliding filament theory. Voila. So we'll go past that. So when they talk about low intracellular calcium, it's the calcium's in the cell, but it's in the cistern. So effectively, it's not within the sarcoplasm. The tropomyosin blocks the active sites. The myosin heads can't attach. The muscle fiber remains relaxed. The voltage-sensitive proteins in the T tubules change shape, and when that happens, the calcium is released. So at higher intracellular calcium concentration, the calcium binds to troponin. Anything binding to a protein we already know is going to alter its shape. It allows that protein influences the tropomyosin to move away from the binding sites, cross bridges form, and it cycles. It will cycle as long as we have energy and as long as we have calcium available to whatever the anatomical endpoint. I mean, like obviously you can only contract a muscle so much until it anatomically reaches its maximum, which is the way it is. And when the nervous stimulation stops, at that point, calcium will be pumped. Important word, because pumping means active transport spending ATP, and the contraction ends. And you're going to see it's not the only part of it. So these are the 4 steps. We're making in the next animation is all about these 4 steps. The cross bridge, where the high energy myosin head. Muscles are always ready to go. In order to release them, you have to impart energy. I can't relax this muscle unless I put energy into it without knowing it. Oh, Lou, you better have some extra energy to get. No, it's automatic, and it relaxes that muscle. At that part of that, as you will see in detail here, involves an ATP causing that myosin head to detach. Okay, so it's already good to go. That's the beauty of the human body. We're prepared in this particular thing for action. So the high energy myosin head, already loaded with energy, attaches once the actin is exposed to it, the active sites on actin and myosin. It develops what they call the power stroke, pivoting, whatever you want to call it, pulls the thin filament toward the midline or M line. Then it detaches, that's what I was just talking about, with ATP, and it'll recock the head and cause the detachment of the cross bridge. Gets the energy from ATP and it's in its high energy state. And they'll show it to you here. It's hard to see. I think to understand exactly what happens, it's not just ATP attaching, as you will see. And where when I do this with the distinction, you may wonder what I did with my AP1 students and you. I just tell them ATP has to attach, but it involves the hydrolysis of the ATP and to change to ADP, and then the movement of the inorganic phosphate. So that's the distinction between advanced physiology and their physiology. So we're good to get busy. Here we go.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position.
Great
As long
slide
as the
here.
binding
Great part.
sites on actin remain exposed, the cross-bridge cycle will repeat.
They don't detach simultaneously, as you will see.
And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
So it gives you an idea of the complexity. So it's not like for A and P, we go, well, the ATP attaches and it separates and the head recocks. But here's a little bit deeper. You would understand better that it has to, once it, ATP may cause it to detach, but to recock it, it has to hydrolyze. When it finally reattaches, look what happens. The inorganic phosphate tightens the bond, and then finally the release of the hydrolyzed ADP allows it to basically fire, ratchet, pivot, whatever words you want to use. So you should memorize at least and try to understand that series of events, because you effectively, you just reverse it going in the other. I detach it with the ATP, okay, and now the inorganic phosphate and the ATP are there. We're constantly, I say it all the time, how much of this stuff we use it when we think. Every time a nerve and a muscle fires, we're constantly resetting it with it. So you have the sodium potassium pumps that are busy. You have what I call double usage here. You need ATP to basically detach it and get it ready for the next cycle, and then you need more ATP to actively pump or transport it back into the sarcoplasmic reticulum, into the terminal cisternae. There's a lot of energy expenditure, which is why I'm so tired just thinking. See, it's harder as you get older. After that fancy dinner we went to, which God bless, my son and my son-in-law were fighting over the check. The minute I said, here, I'll Venmo you the money for the tip. At least they go, shut up, Dad, which I hear a lot. Bless their hearts. That would be a check I wouldn't want to pay. But I digress. But do you get the idea? There's a lot of energy expenditure that's part of it. So it's a complicated process that's there. Aha! What does all this mean? It explains rigor mortis. Okay? So rigor mortis, or it depends on if you're in. A lot of people years ago, when CSI, it was such a big show 30 years ago, a lot of schools were actively, Duquesne probably had the most famous one because Dr. Weck, Dr. Cyril Weck, the late famous pathologist who was involved in the Kennedy assassination, something very, very famous, forensic pathologist and attorney. And people developed a real interest in, what these crime scene investigators do and understanding DNA and things like that. A lot of that came from that show's development. The very famous O.J. Simpson trial was the first time we heard about PCR and RFLP and techniques for all of this. It was the first time people became acquainted with the science, other than people who were familiar with it because at least they understood what DNA and RNA was. Those kinds of things. It was a big talking point. This is 90. That trial started in 95. The murder was in June of 1994, right in the middle. This shows you how long ago it was. The Knicks were in a championship game with Houston in basketball. No basketball fans here. It's been for an ardent Knicks fan. It's been 53 years. But we digress. Nevertheless, it's been a long time. Maybe we got close last time. Not this year. I don't see it. I'm with you. I don't see it. So I was hoping they maybe launch a trade for Giannis, but they ought to. So rigor mortis, rigidity upon death. Why it's so important? Time of death. From a forensic standpoint, you're able to determine that. That's so because that gives you an idea of who had access, assuming that it was not natural causes and there was something suspicious about it. So in any event, people are interested in finding that out, particularly when it wasn't observed that they didn't die at home in hospice or in a hospital. So 3 to 4 hours after death, what happens is the muscles begin to stiffen. That is because the muscles are pre-cocked. The ATP, to relax the muscles, they have to be ready to fire again. As the body begins to deteriorate upon death, that sarcoplasmic reticulum releases the calcium. It leads to rigidity, which typically peaks about 12 hours, and then it will last for X amount of time depending on atmospheric conditions, particularly temperature. And then proteins begin to denature and deteriorate. Okay? So intracellular calcium levels increase because ATP is no longer being synthesized. So once it contracts, no more ATP is going to cause it to detach. So it cross bridges, but because there's no ATP to detach, it stays bound until the proteins begin to break down, denature, whatever word you want to use. And then eventually they are released. So it's interesting, and that typically what they'll do is they'll check the body temperature, the atmospheric temperature, look at the conditions over time. There's more factors than just that. So time of death is really important. Interestingly, stomach contents really play a more pivotal role a lot in this kind of investigation. Certain things, particularly if you have vegetables and things like that, like if you had a salad and certain things like cruciferous vegetables like broccoli, cauliflower, they take a long time to break down. And so it depends on what you ate. If you're having it was breakfast in the morning and I just had toast and coffee, it's gone fast. You get the idea. The other interesting thing is body position. Did you ever hear the term livor mortis or lividity? Lividity means when a person dies, their blood begins to seep out of the blood vessels because it's no longer moving. And capillaries, as you should already have learned, are inherently porous, so blood's going to pour out, so to speak. Let's say if, Lord willing, you fell asleep in bed and you're on your back and they couldn't wake you and that's it. Blood would pool and eventually there will be discoloration on your back and buttocks. Now, if there's discoloration in your back and buttocks and they find you on your face, something sketchy. It might be as simple, we grandma or grandpa, we had to tip them over, we weren't sure and they went on there. Okay, now there's an explanation. But other than that, it means the body was moved and vice versa. If the lividity pattern was here and you find them flat on your back, what happened? Somebody moved that body. So unless there's a reasonable explanation for why that happened, somebody's going to be very suspicious, like the authorities. So that's livor mortis. I'm not going to ask you about that, but I thought you should be. I find it interesting. You've got, you work with the PCR technology in the advanced lab. So I mean, we didn't know anything about that until it came to light during that very famous trial. Even though I'm sure there are more advanced techniques now than certainly there were 30 years ago, but that was the state of the art. First time we heard about that, most of us. Did Dr. Hilwig go into the history of PCR and the person who discovered it and the Nobel Prize was a deceased scientist, gone now, named Dr. Kerry Mullis out in Caltech, somewhere out there, somewhere sciencey in California. He was a case. Brilliant, but when he was like, oh yeah, HIV doesn't cause AIDS. Okay, it's all right. Was something in the air? No, sorry. But it was he was actually, they had retained him on the defense because he knew so much about PCR. So the prosecution couldn't call him because they paid him on the defense, and therefore he was employed by them for that. Very, very interesting. So that is done, that part. And so all of that I've gone over is really relevant, and we're on to that. These next two are not as busy, so to speak. I did want to get through this one because the A PowerPoint with all those animations, because it really highlights a little bit what we're doing today. So this is the idea. When we start talking about muscles and how they contract, and we have, maybe another 10 minutes to discuss it. Whether it's a single fiber or whole muscle, it doesn't matter. The difference is let's say I want to open up my container from the AirPods. I mean, it's not very heavy to lift. But the very first thing you do, even if I'm trying to lift it, is called isometric contraction. It means I have to generate enough force with my muscles. And so before they're moving, same metric length. So we start isometrically, and then once I get it and lift it, then at that point we call it isotonic contraction. Okay, the force has already been there, so the tonicity is the force. Now we're just going to be changing the length. So that's the way this isometric contraction, no shortening, because I haven't generated enough tension or force to overcome the load. I know this is physics. I don't like to do it. The isotonic contraction shortens because we've overcome the load. So we always start isometrically and end isotonically. Both exercises are used a lot, or they were, in conditioning. And it's been probably 30 years since I attended athletic training and physical medicine and rehabilitation conferences. And it has increased light years. So I used to incorporate that, but it's changed a lot. So I don't really want to make a big deal out of it. Because probably the methods are past, I'm past my best by date when it comes to that. So they vary in response to the stimuli. This is about the stimuli. We utilize this, and they still do. For you folks who are in sport, are they using electrical stim on you? Electrical stim is nothing new. That has been around forever. I worked with a physical therapist during my residency who was light years ahead of its time. He was writing articles in physical therapy journals in the 30s about low voltage and electrical stim. It's a great way to, it's kind of one of the labs that we're going to probably do not this week, but next week. We'll look a little bit at the electrical stim aspect of things. Each muscle is served by at least one motor nerve. And the motor nerve contains all of those axons, tons and tons of axons, hundreds, if not more. And those axons branch into those terminals, and we refer to that as a motor unit. Each of which forms the neuromuscular junction with a single muscle fiber. We refer to that as the motor unit. Okay, so motor neuron and all of the fibers, and it depends. If it's small, fine control. I can do this with fine control. Other muscles, not so. Other muscles we don't use for dexterity and things like that, or speech or word formation or facial expressions. We're using them for larger motion. So they're more prime movers in a sense of a physical sense. So the muscle fibers are spread throughout so it avoids fatigue. So we're not, when we want, if this was very light, if this is heavier, I may be recruiting more motor units. And the heavier still, if it was very unusually heavy, maybe this was a block of lead, for instance, that would be more motor units still. So you can see that they're spread out, these different units from different, and you can see there are different axons. And again, there are many coming out all over in different areas. And you can see the micrograph. It looks very much like the illustration and indeed the animations that you saw before. So we can induce, these are laboratory things. When somebody has a twitch and all of a sudden you like that, that's unusual. That usually means there is some type of disorder. It could be a side effect of medication. It could be a neurological disorder. I showed you that nice picture. My first wife was in the picture. She's in a walker now. She takes a variety of medication. She has what they call, you see advertisement, it's called tardive dyskinesia. It probably is a result of medications as well as what's happened to her physically. I mean, she's disabled because of it's difficult. Okay. I mean, thankfully, my kids offer a tremendous amount of support for her, which is great. But it's not, you're not supposed to see that normally. Okay. So, and this is a very, it's just not something from a single action potential we usually see, but we can record it on a myogram. And so a myogram is is a tracing of the electrical activity, and also we can marry that to the mechanical activity. You might remember me doing that in our advanced anatomy lab. I'm going to revisit that next week in lab. Remember when Dr. Buxton came in with his physics for photographing physics class? I had to humor him because I like him. And he's in a better mood now because Liverpool's playing better. And this is consistent. They have a period of latency because there's always a finite amount of time between that signal being generated, let's say from my spinal cord, and getting to that muscle. And that's one of the ways we measure to see if the nerves are impaired. Okay. For instance, if you want to measure carpal tunnel, very, very common. I mean, here's the way we'll put it in really important terms. Years ago, you could operate on carpal tunnels. Oh, they got a carpal tunnel, I'm going to operate. Dissecting for dollars, we used to call it. There were so many operations, the insurance companies went, yeah, you have to have an electromyogram to verify that there is an obstruction to that nerve firing. In order to do that procedure, you need electrophysiological evidence. Ball game change, called pre-certification. So if you're going into fields like that, be aware money plays a pivotal role. So at that point, they will contract and then they will relax. Okay, cross bridge formation, and then calcium going back into the sarcoplasmic reticulum declines. Muscles always contract faster than they relax. Voila. So take a look. So you have the muscle contracting and then you have it relaxing. It contracts rapidly in a matter, in this case, of what? 20 milliseconds? 25 milliseconds? Here's the latency. And then look at the extended period of time, maybe 100 milliseconds. Okay, so we call that an isometric twitch. I mean, it's just a twitch. There's some tension developing that we can measure. Differences in strength and duration of the twitches are due to variations. Eye muscles, rapid and brief. So think how rapidly we can move those up, down, side to side. We do it all the time. Where larger fleshy muscles take calf muscles more slowly and longer. And the really good part about explaining this is this. When you look at the eye muscles, do you remember anatomically the three muscles? They curl together and form the Achilles tendon. So they're your calf muscles posteriorly. The gastrocnemius are power muscles. They're for jumping up and reaching something. The soleus, which is deep and closer to the posterior aspect of the tibia, is a postural muscle. We were standing in line, waiting to get into a show, a movie, a sporting event. Whenever you have to stand in line for a long period, you're sort of like, it's constantly doing with subtle adjustments. But when okay, now it's your turn to go in, Now the activity muscles and power muscles are gastrocnemius. Remember the lateral rectus would be sideways like that, very, very rapid. So it's a really useful chart. I mean, it's still rapid. We're still talking in terms of maybe 100 or 150 milliseconds or whatever. But you see, compared to one another, there's quite a bit of difference. And so we're going to look at the contractions varying. Twitches are either someone who has an abnormal problem, or we see it in the lab. There's something wrong with the muscle, and we can grade the responses based on either frequency or strength. We'll leave it there, and that's what we'll take off, not today in lab, but in lecture on Wednesday. Thank you.